Before we go into the research aspect, let me talk a little bit about my career path. I did my BS in biology, and then I did my MS in biochemistry, and after that I did a second MS in biochemistry and biotechnology. And I was working in a tannery industry, doing leather tanning and processing cow hides for a year, and that's how I got motivated to research. You don't know like why this guy is talking about biotechnology and coming to cardiology aspects. So when I was working with cowhides, I was looking at the connective tissues, and we used to go to seminars where they talk about the connective tissue metabolism and how the fibroblasts are producing the collagen and other stuff. That's how I got motivated to know about more about the collagen chemistry, and finally we could able to attend more seminars on myocardial infarction and other stuff. And one of my senior scientists, he took me to more conferences of all these aspects, and slowly I got motivated to learn more about this stuff. So basically, when I started my career, I have no clue about what I was going to do after 15 years, but it's all like a linear progression, like one thing leads to other. So I started learning more about collagen chemistry, and then I want to have a model. So we created a myocardial infarction model, as my previous speaker spoke about. And then it took us a long time to establish everything in our lab. By the end of my thesis, I had like quite few publications. So I was fortunate enough to get a postdoctoral fellowship at the University of North Carolina, Chapel Hill in 2004. I had a very good mentor, Dr. Susan Smythe. She's a world-renowned vascular biologist, and I have been with her for the past 10 years. Like last year only, I moved out from her. So she's an awesome person to work with. She's a person who can teach you from scratch. And when I joined ENC, I would say that's the best environment for any postdoc to have. Life will be awesome there. I hope it's the same in Yale and Duke, but it depends upon your mentor. So later I will talk about my research, what I did in UNC Chapel Hill. Then I moved to University of Kentucky as research assistant professor, and I pursued my research on vascular biology. And at present, I'm in LSU Shreveport, having my own lab, and still looking at some of the unanswered questions we started 10 years back. Okay, let's go on to the next one. So I will put it in a lighter edge. So by the end of the talk, if we can able to tell two things about my research, I would be happy that I conveyed my research to you guys. So I make it very simple. So here in this slide, whichever it's red means danger. So we are talking about cardiovascular disease, and you can see all the Louisiana, all the way from here up to New York, where you have the people who are working crazy. They are all having a high morbidity and mortality based on heart health issues, basically cardiovascular disease. And the another important aspect is we spend around like $500 billion each year in curing and treating these conditions, and loss of human workforce due to the pathological conditions that exist in our society at the current time. So what leads to this? So here you see our daily menu. When you type junk foods in Google, within a fraction of seconds, 0.34 seconds, you will see our daily menu exhibited. And we never think these are junk foods, but indeed, these are really junk foods, I tell you. So one thing, by the end of the lecture, if you can able to quit these junk food, I would be really happy, like I conveyed my message truly to the audience. So to proceed further, so what happens if you eat these junk foods for a long period of time? When at the age of like 8 years, you get used to this French fries, burger, and all this stuff. And one of the biggest problem in United States right now is the childhood obesity. So we never had, in our generation, we never had this childhood obesity. We had obesity, but we never had childhood obesity. So it's a growing concern, even though people are getting more and more educated, but they think that the modern medicine is well ahead to treat any disease, so they can do whatever they want. So one of the true things is avoid junk foods, do a lot of exercise. So that's the take-home message. But so what happens if you eat a lot of these junk foods? The bad cholesterol, the LDL gets accumulated in your tissues. First, it gets increased in your plasma levels. So that's why you get, when you go to the clinic, they do the plasma cholesterol checkup where they see your LDL, triglycerides, VLDL, and all those stuff. So one of the bad cholesterol is going to be your LDL. When that gets accumulated, it causes a condition called atherosclerosis in your vessels. So when it gets start getting deposited under the endothelial cells, slowly it start occluding the vessel. At one point of the time, the occlusion, the deposited cholesterol is going to rupture. When there is a rupture, when you cut your finger, when you bleed, you apply pressure immediately to stop the bleeding. After some time, if you take your fingers off, you see the bleeding has been stopped, because the reason is platelets comes there and then occlude those vessels and stop you bleeding to death, right? So same way, when it happens internally, when there is a bleeding internal, the bleeding occurs inside the vessel, the same thing, the platelets are like robots. They are going to do the same function. There is a bleeding, we need to go and stop that. So it goes and stop by causing a platelet plug. So by the process, what happens is the vessel get occluded and the blood supply is divided to the distal organs. So that's caused the ischemia of the distal organs, and the rest of the tissue on the other side is going to die. So when you have a occlusion in the heart, you have a myocardial infarction. When you have an occlusion in the femoral artery, you are going to have an ischemic leg. So when you go to the doctor, he is going to perform an angiogram where he is going to inject a dye, and then he is going to see through the radiography and see how far the dye is going through the vessel. And wherever it's getting stopped, then he can see that's the point of occlusion. So then he is going to do a balloon angioplasty by passing a catheter through the femoral artery and passing all the way to the occluded site, and it's going to blow the balloon to relieve the occlusion, and once again the blood flow resumes. So during this process, what happens is when he passed through the guide wire and remove the occlusion in the occluded vessel, the thin endothelial lining, the single layer of the endothelial cell, is going to rupture, and that's caused during the process of clearing the occlusion in the blood vessel. So this is what happens, and once that's single layer of cells, that is the line of defense in the blood vessels, that gets ruptured, the second layer of defense is going to be your smooth muscle cells. And these smooth muscle cells are in the first time going to get exposed to all the mitogens in the bloodstream. It's like when you are driving your car at 80 miles per hour, when you are in the age of, maybe when I was 18 years old, maybe I can do that, not now, after getting married. So when you are driving at 80 miles per hour in the highway, and then immediately a stone crashes on your windshield, and that's it. The whole wind gushes inside the car, and you can feel it. The same way, the windshield is going to be here, the endothelial cells, that is going to protect you from all the shear stress, force, and the blood pressure and mitogen, all the things that's happening inside the blood vessels, and that is protecting the smooth muscle cells getting exposed to that. But when the endothelial cells are ruptured, all this pressure, the mitogens in the bloodstream are going to get exposed to the cells, and if the cells are going to have receptors for those mitogen, and it's going to get bind to those receptors like a lock and key mechanism, and it's going to exhibit its signals, and then slowly the smooth muscle cells, which have been pushed to the edge of the wall, and it has more free space now inside the blood vessel, and it's going to move freely and start to migrate or proliferate based on the signals it receives, and finally cause an occlusion again. So even the patients are relieved for a few years, and later after 3 or 4 years they come back to the doctor, and then he does the angiogram, and he feels like once again the vessel is occluded, but this time it's not due to the clotting of the vessel due to the platelets, but it's due to the proliferation of the smooth muscle cells, and this process is called as restenosis. So one of the aspects we are looking inside in our lab is how to avoid these smooth muscle cells leading to this restenosis. If we can able to stop this, we can able to inhibit the formation of restenosis. So once you have a restenosis formed inside the vessel, the option is you have to do a bypass surgery, or you have to put a stent. You have to put a mesh network inside the vessel to expand it back. So these are the other complications when the patients are facing, but these things happen at the age of like 60 years or 70 years old. So those patients cannot undergo all these vigorous surgical procedures. So we are finding some therapeutic eluents that we can add or we can supplement them so that we can stop these smooth muscle cells undergoing the restenosis after once they have the occlusion or some other stuff. The better thing is to avoid junk food. Okay. So there are a lot of mitogens in the bloodstream like sphingosine 1 phosphate, lysophosphatidic acid, angiotensin, endothelin 1 substance P. There are millions of factors, but we cannot check all those factors, but we can focus our research on one particular aspect and see whether it's going to have its effect on these cells. So one of the aspects we focused on is a simple glycerophospholipid, the lysophosphatidic acid. We use a jargon term LPA in our lab. So by mistake, if I use LPA, please understand that's lysophosphatidic acid. And we wanted to see its effect on the vascular cells. Lysophosphatidic acid mediates its effect through migration, proliferation, differentiation of the cells, and it also causes platelet aggregation, platelet shape change, and vascular cells, the smooth muscle cells and endothelial cells, proliferation and migration. So these things have been studied in vitro. So you can take the endothelial cells, you can add lysophosphatidic acid on top of it, and then you can see what happens to these cells, whether these proliferate or these migrate, and so on and so forth. So we did all these experiments like 10 years back when I was a postdoc. At this time it was a hot topic, and still even now. So we did all those things, and we could able to figure out this lysophosphatidic acid or the LPA mediates its effect through G protein coupled receptors. At that time when I started my postdoctoral work, we had only 3 receptors, LPA 1, 2, and 3, and right now we have like LPA 1 to 6 that are well characterized and 2 more in the pipeline. So we have LPA 1 to 8 receptors. So when we started the study, we had like LPA 1 to 3 receptors in 2004. So as I said, these LPA receptors are G protein coupled receptors that are present outside the membrane in the cells, and once this lysophosphatidic acid goes and binds to these receptors, it mediates different signals, and each and every receptor binding causes different signaling pattern. Like LPA4 causes an binding of LPA to LPA4 receptor causes an inhibitory signal, whereas LPA1 causes a proliferation or migration aspect. It's based on the affinity, like with each and every receptor has its affinity towards the LPA. Like with the lower concentration of LPA, it can go and bind to LPA1, whereas high concentration is present, it can go and bind to LPA4. So based on the amount of lysophosphatidic acid present in the circulation, your signaling effect is going to be different. So these are the aspects we had. So but does LPA play a role in this aspect? So first to know the biochemistry aspect of it, the lysophosphatidic acid or LPA is produced by the enzyme autotoxin acting on the substrate lysophosphatidylcholine. The main source is going to be your adipose tissue, or it can be from your activated platelets. Mildly oxidized low density lipoproteins also contribute to LPA formation, and once the LPA is formed, it acts on the smooth muscle cells or the endothelial cells, and it causes migration or proliferation of these vascular cells, and that's what we wanted to study. So the main question we want to ask is whether LPA is really involved in this process, because there are, like as I said, thousands and millions of mitogens that are present in the bloodstream, and why you want to spend millions of dollars to read about this one particular mitogen. So as I said, we did some in vitro studies and it was promising. So we thought, like, why don't we go over with it? But as I said, we have to have a model first. So how to have a model to test our LPA or lysophosphatidic acid, whether it plays in the vascular remodeling? So what we did was we had a low flow mediated model, which is by ligating the carotid artery. We ligate the left carotid artery here, and after 4 weeks we wanted to see what happens to these vessels, and we compared to the right carotid artery. So what happens after 4 weeks? When we ligate the carotid artery after 4 weeks, we saw a different, completely a change. So this is an unligated one and this is a ligated one, and clearly speaking we can see a difference between the two arteries, right? So to study in depth, if you go and approach a histologist, he will able to differentiate based on the elastic staining. So you have like 3 different layers. One is the outermost layer is going to be the EEL, or the external elastic lamina. The internal one is going to be the internal elastic lamina. The region between the external elastic lamina and the internal elastic lamina is going to be your media. So this is going to be the same in the case of disease as well as in the case of control. But what is the new thing happened is, if you see here you have lumen and then internal elastic lamina, but the lumen is here and the internal elastic lamina is here. So what happened here? So the difference between the lumen and the internal elastic lamina is the intima, or the intima is caused due to the proliferation of the smooth muscle cells towards the lumen, and that's how your occlusion happens. So this is the process we wanted to exactly study how to stop the restenosis. So we had this model in hand right now. So then we wanted to see whether the LPA is going to cause it or not. So what is the aspect, how we can study that? So the approach we had is, why don't we knock out the receptors that is presented for the signaling action of LPA? So if you knock out the LPA receptor 1, 2, 3 or LPA 1 and 2 together, whether then the LPA cannot mediate its effect, right? Then we can subject those mice to this injury model, and then we can see whether the same neointima is happening in these mice or not. If it's happening, then LPA is not playing a role because it's been knocked out already and it's acting through some other mechanism. But if it is going to be not seen in this vessel, then the LPA is going to play a role in that. So that's our main hypothesis. So we generated knockout mice, LPA1 knockout mice, LPA2 knockout mice, LPA1 and 2 double knockout mice with the help of our collaborators. And these knockout mice had some basic defect, like LPA1 knockout mice had craniofacial abnormalities, suckling defects, frontal hematomas, whereas LPA2 did not have any obvious defect, whereas LPA1 and 2 double knockout mice had similar abnormalities as that of the knockout mice. So you can see a knockout mice here and you see a wild type mice here where you can have the craniofacial abnormalities here. It looks so cute though. So we took these mice, the only thing is we cannot subject humans to these models. So we took these mice and we are the boss, and we ligated the carotid artery, and then we wanted to see what happens after 4 weeks. So we did that in these mice. And after 4 weeks, we saw here is our control, SCFT7 black 6 mice, and here is our LPA1 receptor knockout mice. Here our LPA1 and 2 double receptor knockout mice, and we also subjected other LPA2, 4 and other knockout mice. So the pictures which shows like dramatic effects that was exhibited here. So the LPA1 receptor knockout mice had more enhanced neointima proliferation of the smooth muscle cells caused due to the smooth muscle cells, whereas LPA1 and 2 double knockout were partially protected from the neointima formation compared to that of the wild type. Whereas the LPA2 knockout receptors had a similar trend as that of the wild type. It did not have any obvious effect like a hyper or a low neointima. So we could able to figure out that finally there are a cyclic redundancy of these receptors happening between due to the LPA signaling. Like we have LPA1 and 2, it have a cyclic redundancy and it has a protective effect, but you have only LPA1 knockout, it increases LPA3 receptors, and we could able to show by overexpression of LPA3 in these mice and how we can able to delineate its signaling function. And we published this work in Circulation Research in 2008, and I wanted to move on. So there is always an aspect as a researcher, people will ask, like you said that you are passing the guide wire and you denudated the endothelial lining and the smooth muscle cells were exposed, but you said now here you ligated the carotid artery where you didn't denudate the endothelial lining. So we also had another model wherein we can pass the guide wire through the femoral artery, and when you pass the guide wire 3 times through the femoral artery, the endothelial lining will be disrupted and the smooth muscle cells will be exposed to as that you see in the clinical condition, and we saw a similar pattern. So irrespective of the model we had, the neointimal hyperplasia what we observed and the protectiveness we observed was the same. So we could able to convince the reviewer that definitely LPA is playing a role, and we could able to finally show that LPA is going to be a target for this restenosis model. So I moved on with my work because each and every lab has its own baby. Like this project was belonged to my mentor, and then she called me. Then she said that you finished your postdoctoral training. Now, if you want to get promoted to an assistant professor, you should have your own baby. You should focus on your own line of research. Then she was asking what you want to do. So then I said that, okay, I cannot focus on the receptors anymore. I have to find my new line of defense. So as I said earlier, autotoxin was the enzyme that was producing lysophosphatidic acid from lysophosphatidylcholine. So I thought of like, why don't I approach this in a different angle? Why don't I look at the enzyme that produce this lysophosphatidic acid? Or the other option is, I can also look at the enzyme that can able to dephosphorylate or eliminate this LPA from our system. The one enzyme that is going to be present and have an active function is going to be lipid phosphate phosphatase. We similarly use a jargon called LPP here, and for autotoxin we use a jargon called ATX. So I had two options to look at the ATX or the LPP, the one enzyme that produces the lysophosphatic acid, or the enzyme that is going to dephosphorylate or eliminate the lysophosphatic acid from our system. So I took my bet on lipid phosphate phosphatase. Why don't I look at that enzyme? So then I told her, like, I will start working on delineating the LPP role in the vascular tissues, whether it has any significant function to play or not. Then she said, okay, go ahead with it. So as the biochemical aspect of it, the LPA goes and bind to these LPA receptors and mediated signaling, whereas this enzyme lipid phosphate phosphatase, it's going to have its active site projected towards the extracellular space. So it's a 6-membrane domain, and once the LPA is there, it's going to dephosphorylate into monoacyl glycerol and a phosphate. Thereby it is going to disintegrate the signaling aspect that is going to be contributed by the LPA receptor. So if you have more LPP, then it is going to dephosphorylate the LPA, then you won't have any signaling effect mediated in the cells. So I was like having a best confident, okay, it's going to be really fun and it's going to be promising. So we started doing our literature search, and then we could able to find that this lipid phosphate phosphatase exists in 3 isoforms, LPP1, 2 and 3, and LPP1 was coded by gene PPA2A, and the 2 was by 2C, and then we wanted to see what happens to these mice. Already people started working on these enzymes, and they knocked out these enzymes in the mice, and they did not see any obvious phenotypic effect. The mice were normal and they did not see anything LPA getting regulated in this aspect. But when they knocked out LPP3, it was something special. It was not viable, or we did not get any viable pups at all. It turned out to be embryonically lethal. So loss of LPP3 that is coded by the PPAP2B gene turned out to be embryonically lethal due to vascular patterning defects. So it was a good news that, okay, it has a significant effect in the embryonic aspect, but the bad thing is you don't have mice to study anymore. So I was like taken aback. So you cannot perform any more studies now, you are stuck up here. So in the meantime, I was also thinking about, why don't we jump into the enzyme that is producing the LPA. So the producing the LPA is by the enzyme autotoxin. So people have already tried knocking out the enzyme autotoxin, and that one also turned out to be embryonically lethal. So the regulation of LPA is going to be the tight regulation of LPA. The production as well as the dephosphorylation is going to be critical for the normal vascular development. If you are going to mess up the concentration of the LPA in the embryonic stage, then you won't have any viable pups. So we had another option to overcome this. I will talk about that in the next few slides. So in the meantime, Jankanayaki from Japan, he also made some morpholino models, like you can target these enzymes using morpholinos in the zebrafish by blocking the enzymatic effect. Then you can observe what happens to these mice. I'm sorry to say that this video is not working here. If you can excuse me, I can play it through the window media's player here. So this is the control one, where he has focused on the development of the vasculature, and you can see the normal development of the vessels when you have the autotoxin. And let's see what happens. You see the normal branching of the blood vessels, and you start from the base and it goes all the way. Yep.
Manny, I want to let you know the video's not coming through remotely, so we're not able to see it.
Okay, at least LSU kids here, they can able to watch it. I'm sorry about that. I don't know how to put it there. Is there a way to focus?
Maybe Ryan can put the camera on it. That's on you. But
can you focus the camera on the screen?
But it may not play very smoothly. We'll try.
Okay. I apologize for that.
Helps. No, that helps. Thanks.
It's coming now?
Yeah.
Okay, so here is the normal vasculature when you have the autotoxin, and you can see the branching of the vessels. Now I'm going to play another video where you block the autotoxin. What happens? So when you block the autotoxin, the branching is lost and you don't form the normal architecture of the vessel. So it was been clearly shown that tight regulation of LPA is required. Okay, let's move on to our work. So once we have done that, we were looking over, so LPA is required, the regulation of LPA could be altered by autotoxin or by lipid phosphate phosphatase. And as I said, the lipid phosphate phosphatase LPP3 is encoded by PPAP2B gene. So what is the clinical relevance, whether it is, because if it's absent, it's going to be embryonically lethal and there is going to be no humans existing in this world due to the loss of that gene. So why it is important then to study? Then in 2011, a Nature Genetic paper came out wherein they done a study looking at the polymorphism, a single nucleotide polymorphism of some of the 13 genes when they looked over, and they found that the PPAP2B gene was the significant one, irrespective of the traditional factors like high cholesterol, smoking, diabetes. Irrespective of any of these factors, if you have a polymorphism in this 2B gene, you are at high risk. They had an odd ratio of 1.17 with a p-value of 3.81 into 10 to the power minus 19. They did with 85,000 human samples. It's an European study they did, and that's how it puts all our work in our highlight right now. So once they said that if you have a polymorphism in this LPP3 gene, then it's going to be a loss of function in this enzyme, and that's going to cause more LPA action in your vessel, and it's going to put in a coronary artery disease risk status. So now people have started focusing on studying this particular gene. So we are very thankful to that group to show that a polymorphism in this gene might contribute to the coronary artery disease risk. So to validate that further, what we did was we targeted that single nucleotide region where we have the polymorphism with the oligonucleotides, like you can zip it so that the mRNA could not be transcribed. So we did it in cancer cell lines as well as primary aortic smooth muscle cells. When we did that, we could able to clearly show that both the expression of the protein as well as the messenger level and the activity level were significantly dropped out. So if you are going to block, if you're going to have a polymorphism in this gene, then the activity of LPP3 is going to be reduced. If the activity of the LPP3 is going to be reduced, the signaling function of LPA is going to be more, because there is no more dephosphorylation of this lysophosphatidic acid that is going to be in your circulation, that is going to be eliminated much sooner or later. So what are the consequences of reduced LPP3 we wanted to see? But as I said earlier, when you knock out LPP3, it's embryonically lethal. How to overcome that? So you can able to create some research strategies wherein you can able to flux this gene between two LOXP sites. Here the A and B are the LOXP sites I have shown here, A and B. You can insert your gene of interest here, and then you can able to slice it, or you can able to like a scissor, you can able to cut it off whenever you want, like crossing with a particular mice that express an enzyme called Cre recombinase. And this Cre recombinase can be activated by a specific promoter of your interest. For example, in smooth muscle cells, you can, if you want to knock it out only in the smooth muscle cell, you can use an SM22 promoter. If you want to knock it out in an endothelial cell, you can use a Ticre promoter. So you can make these flox mice by inserting your gene of interest between the LOXP site, and you can take these flox mice and you can cross it with a Cre recombinase mice with a specific promoter of your interest, and you can specifically knock out, the offsprings will be specifically knocked out for the particular cell type what you want to study for. So we use that strategy, and then we put our gene of interest between the LOXP site, and then we recombinate. Once the mice is we made the flux mice. We crossed with the Cre-recombinase mice to get our gene of interest deleted in a particular cell type. As I said, we were interested much on smooth muscle cells because smooth muscle cells are the cells that are involved in the restenosis. So we want to specifically knock out with the smooth muscle cells. So we used a promoter SM22 and we could able to specifically knock out in the smooth muscle cells. So the two options we had is whether it will be embryonically viable by knocking out only in the smooth muscle cells, or it's going to be embryonically lethal. Luckily, when we knocked out these in the smooth muscle cells, it turned out to be embryonically viable, and we had a normal blood pressure. But we had changed in the elevated. We had an elevated heart rate, which we will discuss about later. So how you can able to confirm that? You can do a polymerase chain reaction, a PCR. So here you see a wild type band. When you make an insertion of inserting your gene, it will be a little bit of higher molecular weight. So it's going to be in the a little bit above, from 191 to 235. And then when you knock out, it get reduced. It comes to 162. And you might see that there is another band here. It is present. Why it is there? Because you are knocking out only in the smooth muscle cells. So when you take out the tissues, you have endothelial cells, fibroblasts, and other things. When you isolate the DNA, you have smooth muscle cells. That is showing the knockout of your gene of interest. But the endothelial cells and fibroblasts are not knocked out. So it's very specific to the smooth muscle cells. And how you can confirm whether your Cre-recombinase is present? So we can do a PCR for the Cre-recombinase reaction, and you can see that the Tg plus, that the transgenic is being positive here. That's we are shown here. And these top bands are going to be your interleukin 2 as a loading control, because if you don't have a loading control here, then you cannot know whether you have loaded a sample here or not. This will be totally blank. So you have IL-2 present in all the samples, whereas the Cre-recombinase is absent here, and present here in the last one towards your right link. And you have a knockout band that is coming from the smooth muscle cells, whereas the gene of interest is not knocked out in the endothelial cells or the fibroblast. So once we have had these mice, we made our initial diagnosis. Then the first thing we did is we isolated the iota, and we isolated the smooth muscle cells, and we wanted to look for our protein expression that was lost. And then we also looked for the activity assay by mass spec analysis by adding C17LPA to the cell culture, and we wanted to see how fast it's getting dephosphorylated in the cell culture. And we could able to show that among the three isoform, LPP3 is the major one that is present in the smooth muscle cells, and that is causing the dephosphorylation of the lysophosphatidic acid. If you don't have the LPP3, the product monoacylglycerol is not much formed here. So the other isoforms, LPP1 and 2, is not contributing to the dephosphorylating activity in these smooth muscle cells, and it is less present in these cells. So once we have done that, we subjected these mice to the carotid artery ligation, and after 4 weeks, we took and analyzed the sections. When we did that, we saw increased neointimal hyperplasia that is seen in the knockout mice, and here you can see the brown staining for LPP3 that is present in the adipose tissue, both in the wild type as well as in the knockout, whereas the brown thing is absent here, showing that a specific knockout has been occurred in the smooth muscle cells. And the smooth muscle cells are proliferating much higher and faster rate in these mice and causing a more neointima formation due to the less dephosphorylation of lysophosphatidic acid. And we also saw another aspect that there was a positive remodeling, because the lumen was not narrowed here. The lumen was enlarged even though we had a neointima formation. So it was completely a different thing we observed. So we also did other staining for marker for smooth muscle cells to make sure that this is happening only in the smooth muscle cells. And we also did a confocal staining for LPP3, where here you can able to see that the neointima formation is occurring here. So we could able to clearly show that loss of LPP3 is leading to more LPA mediated signaling in the smooth muscle cells, leading to restenosis. And we have not done at one point. We have taken section every 400 micrometer thickness from the point of ligation all the way to the aortic arch, and we could able to show that a clear neointima formation is occurring throughout the carotid artery, and there is a significant positive remodeling. The lumen area is significantly increased in these mice. And further, we also wanted to see what happens to these cells. You can look over the proliferation aspect by looking into some of the signaling aspect, like phosphorylated ERK staining, the extracellular regulated kinase. We did the phospho-ERK staining by Western blot as well as by immunocytochemistry. We could able to clearly show that these cells are still in a proliferating phase even after 4 weeks. and the inflammatory cells were higher in these knockout mice. The inflammation macrophage staining was higher in the neointima area, as well as we saw the interleukin 6 expression was still higher. So I won't talk much about that. We have explained in-depth analysis of all these things in our paper that is recently published in 2013. And the take-home message is when you have a single nucleotide polymorphism in this gene, it leads to coronary artery disease risk. And when you knock out LPP3 in the smooth muscle cells, it's lead to more proliferation and vascular inflammation. So that we can claim the fact that LPP3 act as a negative regulator of the vascular function. When you have a loss of LPP3, you are going to have a more deleterious effect. So the future aspect, we are focused, as I said, when you eat junk food, you have cholesterol deposition. So we have another model, like we can subject mice to atherogenic diet, and after 16 weeks, you can take the aorta and strain for the atherosclerotic plaques. When we did that, we saw a significant accumulation of LPP3 in the atherosclerotic plaques, and we also did immunohistochemistry in these mice. And we could able to clearly show that clear localization, the yellow color, what you see here for the LPP3 with the smooth muscle cells, a clear localization is happening here. And I won't talk much about what happens to loss of LPP3 in the endothelial cells, but I will give a clue. When we knocked out LPP3 in the endothelial cells alone using a Tycre promoter, it turned out to be embryonically lethal. And we have to overcome that effect by a tamoxifen inducible model, which my student Megan will be presenting in the LBRN poster. So fascinating work. Why it is important? Because the future is going to have more like a personalized medicine. You cannot have one small one same drug that is being prescribed for everybody. Somebody might have a polymorphism towards this gene, then they need an extra care. They have to be treated for the specific mitogen. That's why it's these researches are important. So the future aspect is going to be based on more personalized medicine. Nobody can be given like, okay, we can give resveratrol or clopidogrel drug for antiplatelet or antithrombotic effect. If you don't have an LPP3, then your LPA is going to be high, your platelet is going to aggregate in a much faster rate, and your smooth muscle cells are going to proliferate in a much faster rate, and your plaque formation is going to be higher. Then you have to inhibit those signals by blocking the G protein coupled receptors. You are going to block the LPA1 or LPA4 or LPA1 and 2 together. Which is the best one? You have to figure it out, and you have to formulate your personalized drug therapy to the patients. That's what our future is all about. And last but not least, I thank all my team members and my collaborators throughout the globe. And last but not least, my fundings from American Heart Association and NIH for exploring this new pathway. And I right now will take up any questions from you guys. Thank you.
Start with New Orleans. New Orleans, do you have any questions? Just chime on in. Again, wave at us so that we know you're. Yeah, thank you. Okay, good. Well, Shreveport, any questions from your local audience?
So my topic did not make any sense, or it made a lot of sense. You guys understood everything. I can take it in two aspects.
Baton Rouge, any questions? I got the mic. Over to you, Bill.
Okay, being the last lecture, I won't trouble you guys a lot. I hope you all had a wonderful research training in all the labs. Hope to see you guys in the poster session then.
Great, thanks.
Thank you.
I actually have a question from one remote student.
Sure.
She's asking, you mentioned something about LDC. What does it stand for?
LPC is lysophosphatidylcholine.
LDC. Is that what you said? Is that right, TT?
I expressed LDL, low density lipoprotein.
At the beginning, something about LD, like in David, C. Somewhere at the beginning of your slides, she said.
The lyso-PLD?
Yeah, unfortunately, I don't have audio from her, so.
Okay. Sorry about that. Maybe I can meet him on the poster session. I can clear his doubts.
Yeah, so she's in Natchitoches, and that's
Okay.
where she's. So she should be there.
Great, thank you.
Thank you. Thank you, guys.